I am Gaia Pigino, and I currently am a research group leader at the Max Planck Institute for Molecular Cell Biology and Genetics. I'm having some problem actually changing slides in my presentation. I'm gonna.
You have to use the trackpad, not the button.
Okay. Let's see if it works. Yes, thank you very much, Alessandro. Yes, so I obtained my PhD in Italy at the University of Siena, Department of Evolutionary Biology, where I also did a short postdoc. During this time, I learned the fundament of electron microscopy and tomography. In 2009, I moved to Switzerland, first to ETH Zurich, and then with an EMBO fellowship to the Porsche Institute, where I work in the lab of Takashi Ishikawa, and I specialize in cryo-electron microscopy, and more specifically in cryo-electron tomography. In 2012, I moved to Dresden, to the Max Planck Institute for Molecular Cell Biology and Genetics, where I started my independent research lab. And there, thanks to the very well-equipped facilities of the CBG, I was able to expand the set of techniques and technologies and methods, and so that now my lab is not only relying on electron microscopy, but we can space from biochemistry, in vitro reconstitution systems, and light microscopy, and also correlative light and electron microscopy. So I will join the Human Technopole at the beginning of 2021. Where I will have a dual role. I will, of course, lead my research group, and as the associate head of the Center for Structural Biology, I will be strongly involved in establishing and developing the structural cell biology part of the center, which is related to a number of technologies, very new technologies. As you might know, the cryo-EM field has been revolutionized in the last years by a number of innovation in the technologies such as cryo-fib-EM or cryo-correlative light and electron microscopy that allowed us, also at HT, because we will have all these technologies at HT, allowed us to investigate the molecular structure of large cellular machines in the native context of the cells. So it really allowed us to investigate processes that happen in the cell at a structural level. So from a more personal scientific interest, I am very much interested in understanding how simple molecular components self-organize to form large, complex cellular machines or organelles that are fundamental for the function of cells and also fundamental for organismal survival. And in my lab, we address this kind of questions by investigating a particular organelle that is the cilium. The cilium can be, it is an extremely conserved eukaryotic organelle. You can find cilia in single cell organisms such as green algae that you can see there in the video in Chlamydomonas. You see these two elongated structures. These are the cilia of Chlamydomonas that allow the cell to move, but also allow the cells in general to sense the environment and to send signals or to receive signals. Now, these are multitasking cellular machines, and the structure of these machines, cellular machines, are fundamental for the function. So defects in the assembly and in the structure of cilia can generate a number of devastating human pathologies. So it's very important to understand the structure, and it's very important to understand the mechanism of function of these organelles in order to understand these human pathologies that are collectively called ciliopathies. So my lab in the last year contributed a number of discoveries in the cilia biology field. We mostly focus our attention on the molecular bidirectional transport that is required for the assembly of the cilium. And in the future at the Human Technopole, we will want to continue investigating cilia, investigating the high resolution structure of the intercellular transport system, so this bidirectional motility system, its mechanism and regulation. In a second line of research, we will want to investigate the role of tubulin post-relational modifications, also known as the tubulin code, in regulating cilia assembly, in regulating the transport, which is also fundamental not only for the assembly of the cilium, but also for the function of signaling of the cilium, and also in regulating the beating of the cilium that allows motility of cells. And this particular project, long-term project, is already financed by a consolidated ERC grant that I just achieved last year. Third, but not last least of the list of my research direction is investigating primary and motile cilia in human disease. So I must tell you that most of the research that is done on the structural characterization and mechanistic understanding of cilia is done on cilia that belong to single cells organisms such as green algae like Chlamydomonas. This is just for technical reasons a much more accessible system. However, this results in a lack of information about molecular structure, composition, and mechanism of function of the cilia that are maybe very important for us. So cilia that are present in tissues and organs in our body. So one important direction will be to develop methods and also in order to investigate this kind of cilia. And for all this project, of course, I will be happy to collaborate with labs abroad. I'm already doing that, but also labs, research groups in Italy. And in particular, for the primary and motile cilia in human disease project, I will be happy also to interact with clinicians and hospitals in the future in Italy. So the projects I propose are challenging and they require interdisciplinary approach. And this will be a very strong point at the Human Technopole, because having the different centers and having strong facilities that will be accessible to all the researchers, we will be able to, not only generate synergies, but be able to use different technologies that will allow this kind of projects that require an integrative approach. And so beside that, I would like just to mention that the technologies that will be available for the structural cell biology at the Human Technopole are what we call cutting edge technology. So these technologies are very new, and therefore they also require development not only of the technology and hardware, but also development of new methods that will allow us to get the best out of them. Methods that do not involve only wet lab work, but also methods that involve analysis of the data that we get. So collaborations, for example, with the groups that can provide new algorithms and image analysis strategies will be fundamental to get the best out of our images. And with this, I would like to thank you for your attention, and I'm happy to see you in person in the future, hopefully, and to discuss science with you.